All right, section 4 time. We're going to shift gears from viruses to prokaryotes, so bacteria and archaea. Our goals for section 4 is to know all the vocab, as always. Not that vocab is everything, but it's a good start. You need to know it. Second thing is to understand the role of prokaryotes on Earth. What do they do? Why they do it? What would happen if we didn't have them? They're very, very important. Compare and contrast bacteria and archaea. So how are they similar and how are they different? Because when we look at them under the microscope, they kind of look the same. They're prokaryotes, they are single-celled, no nucleus, things like that. But molecularly, they are very different. I need to know how. Last but not least, what are common bacterial survival strategies? So bacteria are very good at surviving. How are they doing that? Let me know. So prokaryotes are widespread on Earth. Yes, they are, right? There's like 7 billion people, maybe getting closer to 8 billion people on the planet right now. Yeah, where there's this many individual prokaryotes. That is 10 to the 30, so 10 with 30 zeros after it. It's a lot, like bigger number than humans can actually think of in our heads, right? So they're everywhere. They're on everything. They're living inside everything. They're in awesome, cool places and like really dangerous places. They're just everywhere. Rocks, deserts, hydrothermal vents. They might even be out in space. We haven't found one yet, but that'd be really cool. Now we classify prokaryotes based off of their relationship with oxygen, and there are 3 classes I need you to know, right? There is first the obligate anaerobe, right? So obligate kind of means like you're obligated to do something, right? You have to do it. Well, they're obligate anaerobes means they are anaerobic. They do not like oxygen. In fact, they will die if exposed to oxygen. So obligate anaerobe, they are poisoned by oxygen. And this would encompass things like the extremophiles we learned about a couple of chapters ago that live in like super salty places or super hot places or super cold places. Others of them live in digestive tracts of organisms in a symbiotic relationship. So the prokaryote will help digest the food of whatever organism host it is in, and the host will get access to those nutrients and provide a stable place to live. So a warm, safe, nutrient-filled place for the prokaryote. The second kind of prokaryote I want you to know is an obligate aerobe. So if an obligate anaerobe can't use oxygen or can't be in oxygen at all, obligate aerobe must have oxygen. Right? So just like eukaryotic cells, they require oxygen to survive. And these have some of the most dangerous pathogens in the group, like tuberculosis, which is a really nasty infection that gets in your lungs and causes you to attack your own lungs and cough up blood and die. If you read Edgar Allan Poe, you'll know all about tuberculosis pretty quickly. Now, the last one, and the coolest in my opinion, is the facultative aerobe. So these boys, they don't care. Right? They could be in the presence of oxygen and not die, or they could not have oxygen and still not die. Right? They can survive with or without oxygen in the environment. So some similarities between bacteria and archaea is, of course, that they're both prokaryotic, single cell, they don't have nucleus, membrane-bound organelles, things like that. They can have these special DNA segments called plasmids that are small and circular and outside of their main DNA chromosome. They can replicate separately, they can even be traded, and we'll talk about that later on. Now, many bacteria can move using things like a flagella, and flagellum is Latin for whip, so you're literally like whipping around to go places, which is really, really neat. And they also have little things called pili, which are little tiny hairs that stick out. So think of flagella as like the big one that moves them a lot. Well, pili can move them very little and helps them to grip onto surfaces. But the interesting thing is that archaic flagella and bacterial flagella aren't made out of the same substances, but they still have the same general structure. Now, the differences between archaea and bacteria are just as numerous as the difference between archaea and eukaryotes. So think about that. So you and I are both eukaryotes, and there are just as many differences between us and a single cell prokaryotic thing that lives at the bottom of the ocean as that thing at the bottom of the ocean is with bacteria just roaming around. So they're very big differences, and lots of that is going to be in the lipid membrane. right? So archaea have this very unique lipid found in their membrane, not found in any other organism on the planet, right? And that's why it had to be split off way back in the day. Bacteria have this special substance called peptidoglycan, and even within the presence of peptidoglycan, there are two classes of bacteria. There is the gram-positive bacteria, which has this really thick layer of peptidoglycan, and the gram-negative bacteria, which has the relatively thin layer of peptidoglycan. Now, for survival strategies, bacteria who normally reproduce asexually have a way to increase their genetic variation, which it used to be thought of just as a sexual reproductive advantage, right? So how they do that is by doing this thing called conjugation. So in conjugation, two bacteria can link up together through a tube made out of the pili we talked about before, right? And through that pili, they're going to send the plasmids through. So it's almost like a weird bacterial telephone call, like, hey, do you want this plasmid? It gives you resistance to this antibiotic. And the other bacteria is like, oh, heck yeah, I'll take that. I'm not resistant to that bacteria right now. And what you have at the end of the day is two bacteria that have that plasmid when it used to be just one over here. And that's really different than the normal way of reproduction, right? Which is just asexual binary fission. Well, in this case, this is a kind of type of sexual reproduction. They're trading genetic information that's different and unique. They now both have it. It doesn't work necessarily the same as eukaryotic organisms, but they did trade and increase genetic variety from that trade. So it's really neat. We can look at microscope images of these conjugation bridge, the tube, going between the two bacteria. It's really neat to see. Now, second survival strategy is how bacteria can go off and, survive in extremely dangerous places or in places where there's not a lot of nutrition for them and for very long times. And that would be called an endospore. So an endospore is when a bacteria copies its DNA and makes a thick coat, thick wall around it. All right, why would they do that? Well, it's kind of like a time machine, time capsule. And what the endospore is waiting for is for conditions to return back to being favorable, where that means the presence of water, where that means it's not so hot anymore, not so cold. I mean, these things are crazy. Right? They have even been found on Egyptian mummies, which are thousands of years old. So that tells you just how effective the endospore is. Very, very effective at protecting the bacteria. And it works like this. Right? We have a bacterial cell. They're going to copy the DNA. And inside that, instead of splitting across like it would be in binary fission, they're going to start wrapping that extra copy of DNA in that thick coat. And the coat is going to get thicker and thicker and thicker until it actually has a couple of layers. And notice that like the rest of the cell kind of gets broken down, right? All that's left is this. But we see how effective it is. Like the original cell might have gone and died, but everything the bacteria cell needs to come back to life is contained within this endospore. And whenever it has favorable conditions, it just starts right back up. And it's very good for the bacteria. Like I said, on mummies. They've been found on mummies that have been sitting in a tomb for 3, 4, 5,000 years. Okay, so in review, how do we classify the prokaryotes based off their relationship with oxygen? What are the 3 ways? Compare and contrast the similarities and differences of bacteria and archaea, paying special close attention to their molecular makeup. How can bacteria increase their genetic diversity? And what is the purpose and point of an endospore? Why would a bacteria want to do that? Okay, if you have any questions, please don't hesitate to email me or contact me on the Google Classroom. And as always, thank you for watching, and I will see you in the next recording. Talk to you later.